All right, I think we'll go ahead and get started with our second speaker this morning, Dr. Rohit Bhargava, who's a professor in bioengineering here. And he's going to be talking about chemical imaging for digital molecular pathology. With really the goal to be able to take some of these biophotonics, these imaging techniques, spectroscopy techniques, and better understand what's always been a very subjective art of pathology.
Thanks, Steve. Thanks. Hi, good morning, everyone. There's no response back. Usually I get a better response than this in my classes. But we can try this again. So good morning. Wow. Thanks. Well, I hope you're all having a good time at the summer school. This is a lot of fun, right? So what I'll try to do is relate back some of the sort of physics you might have learned, some of the other techniques and imaging approaches you might have seen before, to work that's done in my lab to some extent, and then we'll try to expand out broadly as to where this might all be going. So before we begin, let me just introduce a couple of clinical problems, right? So a part of my lab is motivated by thinking about cancer, right? So in this country, at least, if you look at the cancer healthcare enterprise, the way it turns out is we take perfectly normal people, take people who have no symptoms, tell them go to the doctor every year, go for a mammogram or a PSA test or whatever. So we put them through a screening approach. And at the end of the screening, we just want to say, is there something suspicious, right? The it here is not the person. The person may or may not be suspicious. There's something suspicious in their test, right? That's the idea. If there is something suspicious, which is about 1 million people, for example, in prostate, 1 million in breast in the country roughly, we take a needle, put it inside the body, take a small piece of tissue out. The small piece can be anywhere from 100 microns to a millimeter, can be anywhere from a couple of samples that are directed by ultrasound to basically putting a pincushion of needles, 46 needles in the prostate, because there is no ultrasound to imagine where the tissue is, right? So there's a fairly, I would say, almost primitive analysis at this stage where you just blindly go and take some things out in some cases. And even in cases where it's directed by imaging, you can't really see locally. So oftentimes you sample multiple times and so on. So 1 million people, anywhere from about 5 to maybe 50 to 100 samples per person at this stage. Then somebody has to look at all of that and come up with a diagnosis. And they have to say this is cancer or no cancer to begin with. If it is cancer, then what they're interested in is what's the grade. Has it reached outside the organ? Will it actually metastasize? Will it go outside the organ, or will it remain confined? And if those kind of estimates can be made, then perhaps the kind of therapy can be determined, okay? And therapy, depending on which stage and grade and all that business, can range from the low several $1,000s per month to the upper $20,000, $30,000 per month sort of range. So these are some very, very important decisions here. And finally, a lot more people today are surviving with cancer than have ever before. So the question here, again, the numbers of people expand out so much, and every single recurrent case is actually very dangerous because most of the deaths due to cancer are because of recurrence, right? So you can get cured, everything's fine, but then you need to be monitored pretty frequently to make sure actually things that come back which are more dangerous aren't really occurring. So notice a few things that are going on here. The number of people is immense here, and the tests are fairly routine, right? So you go into the doctor, you get a PSA test, you leave, and so on. The dollar amount per test is not a whole lot. The number of people is very large. When you go down here, the dollar amount goes up dramatically, the number of people goes down dramatically, and so on. So it's quite interesting, and I wish you would take this on as a Google assignment for today, is go and multiply the number of patients by the dollar amount per patient, and you will find that it is unfortunately constant at all stages of this disease. So every specialty makes an equal amount of money in this business in some ways, right? That's one way to think about it in engineering terms, and which is quite surprising. So if you were to disrupt something here, now imagine what this implies for biophotonic technologies or for imaging in general. If you were to disrupt this curve, if you were to disrupt the status quo, what would you do? And if you disrupt the number of people, then your dollar value per test needs to go up, or that specialty needs to disappear. And those are really also the challenges beyond technology to adoption in this case. Okay, so let's discuss technology first, and I'll tell you how that paradigm will be changing perhaps in the coming time. So the technology for this middle portion is quite easy. If you are biopsied, then a piece of tissue comes out, you slice it thin, put it on a glass slide, stain it with a specific set of dyes. In this particular case, this is a fairly non-specific dye combination, hematoxylin and eosin. So all the protein-rich regions are stained pink, all the nucleic acid-rich regions are stained blue, and that's the contrast you get. A pathologist typically looks under a microscope for this, and you actually don't even need a microscope in this particular case. Notice how there's a thickening of the tissue here. It's sort of less transparent in that regard. There's a lot more stuff going on here. There's a lot more stuff going on here. So these sort of the first thing a pathologist would really do is look up, under the lights and see if there are gross regions like this where there's like clumps of tissue. And you actually don't even need a microscope. You can use your eyes and say, well, something's not right here, something's not right there, something's not right there. And they used a sophisticated microscope and actually figured out here's cancer, here's cancer. This, for my benefit, they said is normal, right? So we're trying to do exactly the same thing in our lab, except we don't stain tissue. This is unstained tissue. This is one section of it. This particular tissue is just the next section. Okay, so it's the next cut. It's the next 5 microns section from this. What we have done here on the left-hand side is actually color-coded the tissue for different cells. So the green are all epithelial cells. The pink are all the non-epithelial cells, which we collectively call the stroma. The reason is because this is prostate tissue, and almost all cancers in the prostate arise in the epithelial cells. Okay? So what we're going to do is identify where cancers can possibly arise, and then computationally suppress everything else. So on the image in the right, you see all the pink stuff is gone. We're only left with the green pixels, and then we analyze the green pixels further and just leave them green if we're sure that they are normal. If we're not sure that they're normal, then we color them something else, right? In this particular case, we've colored them with grade 3, grade 4 cancer in the prostate. There is no other cancer in this. So our diagnosis would be that there's a lump of tumor here, there's a lump of tumor there, there's a little focus of tumor down here, and otherwise it's normal. So we go back, and our pathologist indeed confirmed that they missed this little section of tumor there. There's actually a little bit of tumor there. They were quite happy seeing this, and those are the 2 major foci. To be fair, you don't need to look much beyond that. But again, you notice right away a couple of things that are happening, right? So in practice, you're rushed for time. You're doing this manually. You're making decisions with a felt pen, right? You're going there and circling things. Not very precise, quite subjective. The chance in your rush or in the speed to get things done, you might miss something and so on. So automated technologies like this, and I'll show you how exactly we do this, but if you had a tool that was able to do this, what you would be able to do is put your slide in, get a color-coded map, and your pathologist can now come in, and you're not going to miss anything on this, right? In fact, you can go one step further. It doesn't take a genius to do this. You can get rid of all the green color as well, and just look at the yellow color or purple or what have you in this image, and you can point that out in this thing. So it's this idea of using new kinds of imaging to give you new kinds of visualization really is the key behind what we're going to talk about today, right? The second thing, which sort of was implicit in what I was saying, is what we are doing is replacing this sort of manual staining approach with computation. So there's a computer behind it that's allowing us to make these visualizations, which otherwise required a microscope and some dyes and stains and so on. And the whole idea here is that instead of doing staining, sectioning, microscopy, human analysis, and then come up with a decision, it's all done in one step, right? The microscope itself is the human, or itself is the decision maker. The microscope itself is the stainer in that sense. So instead of doing microscopy as a module in the diagnostic process, the whole thing can be done together in what we call smart microscopy in this way. Don't worry about it too much. This is only for grant writing purposes. Otherwise, it doesn't mean a whole lot, right? But basically, again, think of this idea as, and we'll come to this point again later, think of, for us who are more engineering inclined, perhaps here, who are more interested in the science, think of this as a merging between computing power and optics, right? That's the best way to look at it. And it will merge to a point in the coming slides where you can't really distinguish one from the other. And in fact, what we're doing is playing one off the other, so you actually have a much better synergistic system than you would have alone either way. But the second thing, which I'll show you in a few slides to you next, is that the way we're doing this, how we're getting this image, is not dependent on actually the morphology of the sample, as most imaging techniques and as human recognition is dependent. It's actually dependent on the underlying chemistry. in your sample. Okay. So what I'll show you in the next few slides is that in these kind of images, every pixel actually has an associated spectrum with it. And the spectrum could be infrared or it could be Raman, which are the two modalities in our lab. Again, there's a whole world out there. This spectrum could be a mass spectrometer-derived spectrum, for example. It could be near-infrared. It could be other kinds of spectra. It turns out that in the infrared and Raman, as you heard in the talk last week, the fundamental vibrational modes are measured. So fundamental molecular changes are measured, or fundamental interactions are measured. In the near-infrared, for example, overtones and combinations are measured, so the information is much weaker. In mass spec, you only measure proteins. You don't measure nucleic acids. You don't measure lipids. You could, but you don't measure them normally. You're more sort of protein-focused. So infrared and Raman are perhaps the only techniques which allow you the full chemistry of tissue to be obtained. They also allow you to obtain that non-destructively for the sample, right? You still have to section the sample in some cases. You could do 3D, but you have to section it today to get the best information. So once you have the sample, it doesn't destroy the sample like mass spec. So those are the advantages, and that's why in our lab we sort of restrict ourselves to infrared and Raman, and I'll tell you that. But again, the hypothesis is that the chemistry plus structure is better than just morphology. That's the idea, and we'll try to show that. So let's take a look at how this actually works and what are some of the important questions here. So this is a chart showing you, the sort of distribution or the health impact of prostate cancer, right? And these are roughly the kind of people who are, this is schematically showing whatever is written on this slide, actually. So if you look at about 1,000 men screened over a 10-year period, right? That's what we are doing in this case. Over a 10-year period, out of the 1,000 men, 100 to 120 will get a false positive diagnosis of prostate cancer, right? So they have no symptoms whatsoever. You stick a needle, pull a piece of tissue out and say there's something wrong. And it turns out the healthcare enterprise was wrong in actually telling them that you have cancer. So about as equal to the number of people who are diagnosed with cancer, we have a number of people who are just because of the side effects of testing are diagnosed with cancer and they don't really have cancer. Right? Of the 110 who actually are diagnosed with cancer and who do have cancer, right, at least 50 will have treatment complications of some sort. So they'll be incontinent, they'll be impotent, perhaps for life, perhaps for at least 6 months, 2 years beyond their treatment. And so it's a pretty horrible price to pay. 4 or 5 will die despite us doing all of this business. 1,000 people screened for 10 years, right? That's a fair effort. 4 or 5 lives will still be lost from prostate cancer. Despite doing everything we can, despite 97% of people in this country going in for aggressive therapy for prostate cancer, we save 1 life for screening 1,000 men over 10 years, right? So these are some of the sort of shocking statistics. So the trouble with prostate is not the fact that 4 or 5 die. You have to die of something, I'm sorry, but this is, it's unfortunate that this happens. But look at the price to prevent this one death. It's this, and that, right? So there's a lot going on in the prostate world right now, which is somewhat controversial. Whether should we screen these people or not is a controversy. How should we treat people or not is a controversy, right? And it turns out once you have a diagnostic test, it turns out you can't go back to people and say, no, don't get screened. We just told you last year that you should go in and get screened every year. And this year we're telling you, no, the cost is going up. We're kind of uncomfortable with you getting screened. So I think the cat's out of the bag. There's nothing you can do to prevent prostate screening or breast screening for that matter now in the country. But what you can do are provide technologies that very rapidly tell you that these 100 or 120 are not in trouble, right? So, okay, we biopsied you, sorry, but go home, there's nothing to worry about, right? We tell them very quickly. What we can do is tell these 50, look, there's nothing really going to happen to you, but you shouldn't really go in for surgery because you could be incontinent for life, right? Then it's their decision. For these people, we have to say, sorry, there's bad news, right? Despite everything we've got right now, whatever we do right now, there's nothing we can do to help you. And for this person or these people, I don't know what between 0 and 1 means, but for half that person, I guess, we have to tell them that, look, there is a therapy, go in for this surgery, do this, and everything's going to be fine in this case, okay? But the trouble is, and here's the catch, we don't know who falls into which group, right? So if you take one person, if you take an individual, you don't know if they're here, there, or there, right? We have absolutely no idea. We can distinguish between this group and this group somewhat well, but we can't do anything better than that, okay? So what I'll try to show you towards the end of the talk are some of the newer methods that are allowing us to definitely distinguish this from this, without a doubt, at 100% accuracy, but also perhaps to pull out 40 out of these 50 people maybe, or 35 out of these 50 people, and say you don't really need aggressive surgery, don't go there. We're far from identifying this one person yet, but we hope to get there with, I'll show you how we'll actually get there. Okay. So in my lab, we're trying to tackle these problems. The first thing we do is, can we have a technology that goes and tells people that, look, here you've got 100 cases of prostate. We can do this very rapidly, and we don't need to spend human time in looking at biopsies. The next sort of case comes in distinguishing this group from this group. Could we diagnose whether there's a cancer or not? And can we do that with very, very high accuracy so that we can actually replace a human in some case, right? So in this case, it turns out that you need about a 1% accuracy, 1% error rate, right? Anything above that gets you into a territory which is uncomfortable for humans. Okay. If we take this to the next level, could we actually diagnose so we can get more information here, right? So could we figure out who these 50 are, and can we do a good job of that and this? But also, quite importantly, can we take the best pathologists in the country, have their expertise matched by this computer program and this microscopy, and bring it down here, right? So we have the best technology, best sort of capability in this case, and it's available all over the world, for example, even here and others. And then finally, this is perhaps the most pressing need in prostate today. It's not the fact whether you have prostate cancer or not, it's what's it going to do. Is it going to leave the prostate and go to your bones, which is really, really a bad thing, or is it going to stay in the prostate until you die of old age, right? That's the key. In prostate, for example, if you're 70 years old, you have a 70% chance of having prostate cancer somewhere in your prostate. If you're 100 years old, there's a 100% chance you have prostate cancer somewhere in the prostate, right? So the question is not whether the prostate cancer is present in the prostate or not, is whether it's going to escape from the prostate and go to your bones and actually kill you in that case. In this last category, we are severely lacking in the clinic. We just have no idea how to predict the sort of medium range, right? So if you're at the fringes, if you have a very low-grade disease, your PSA is not very high, then we can safely say, look, there's not a whole lot of problem. We can perhaps operate on you, next year or a few years down the road. If you have high-grade disease, if you have grade 5 or something, your PSA is 60, 70, 80 nanogram per ml, for example, then you know this is a very dangerous cancer that's growing rapidly. We need to operate on you today, right now, right? But only about 10% of people fall into that category, right? We've become so good at diagnosing people now because of screening that most people, 70 to 80% of people, fall into this middle category when their cancers are diagnosed. So they have grade 2 or 3 or maybe 4, even 4s are quite rare. Now their PSA is between 4 and 20, for example, in this moderate range of grade. And the trick is, what do you do with them now, right? So for 80% or 70% of prostate cancer patients who are diagnosed today, we actually don't know what to recommend in this case. Should you wait for surgery, or should you go in right now? Should you maybe perhaps see if it changes or not? There's nothing we can say confidently at this point. Okay? So what I'll do is go through, first explain the technology, what it is that we're doing in the lab. Then I'll go through and give you examples in each one of these 4 categories and see what we've done. So we've been pretty successful at this and this. I mean, these are somewhat easier problems. We're almost at the point in this where we can do things quite reliably. And this, what I'll show you is some studies that we've recently done, which definitely beat the state of the art that's available today in the clinic, but they're not quite there where we would like them to be. Okay, so this is just a couple of, just to get you oriented also with what you're going to look at. This is the aforementioned H&E you saw on the previous slide, right? And what you'll see here are images I'll show you that look like this. So these images are color-coded, and there'll always be a key on the side. So if you want to figure out what color is what, the whole presentation has consistent colors. So if you put green epithelium in your mind, then every slide, the green, you can just check off it's green epithelium, okay? So the whole thing is consistent. You'll see this, you'll see a length scale, so you'll see how big the tissue is, and you'll also see whatever test we're doing will be right over at the top. Okay, so that's how you'll look at this. Now this study is a historical study now in the sense that it's really old, but it's also very interesting. This study really launched the sort of time period where this kind of analysis was possible. And what we did here was, this took us almost 3 years to acquire the data. And what we did here was took 8 samples from a single patient, a second patient, third patient, one clinical setting, second clinical setting, and so on. And what we could show here is that the accuracy with which you could classify these things was A, very high, 2, it was robust and reliable, and third, it was present, it was doable across multiple clinical settings. And fourth, it was doable over large number of samples, right? So when you read literature like this and you look at people claiming to do spectroscopy of this and diagnosing that and so on, I think a few things need to be looked at. What is the variability of the sample set that they were getting, right? Was it from one setting? And there's a certain claim you can make from one setting, right? You can say, look, there's perhaps potential in this. You can't say you have a diagnostic test, right? How many samples they looked at, how many spectra they looked at, how many were in the training set, how many were in the validation set, and so on. So all of these issues will come up. But you'll see these numbers over here, and then we'll try to quantify the accuracy a little bit more also on the next slide. So what's happened in the intervening 10 years is essentially this. So this is some data from last year. And what we've done here, that was prostate in the past. This is colon histology. And what we're trying to do here is only figure out where each cell type is, right? So it's not particularly complicated. We're just trying to say, can we find different cell types in this? And again, there's a color key here. But notice this new curve that has shown up here, and I'll explain what this is. So anytime you have a diagnostic test, you're trying to map a pixel onto its identity, right? You say this pixel is a blood vessel, for example. You're either right or you're not, right? If it is indeed a blood vessel and you're right that it's a blood vessel, then your sensitivity is high, right? You're accurate. If it is not a blood vessel and you correctly said it's not a blood vessel but it's something else, then you're highly specific, then your specificity is very high, okay? So oftentimes in biomedicine, the sensitivity specificity trade-off happens constantly all the time. We just never know it, right? So if you take immunohistochemistry, for example, and you stain something, right, you can be very specific. But if you use too much stain, then you're not particularly sensitive because the analytical background has swamped out your signal, right? So inherently, there's always this balance between sensitivity and specificity. In immunostaining, you just never talk about it because it's never possible to really quantify it, right? And there's certainly in immunostaining, I think the primary source of error is batch-to-batch variability. So you get a new antibody, it doesn't work somehow magically, or it didn't work quite as well as that. You treat the tissue differently, it doesn't work quite as well anymore. What we can show here is it doesn't matter how you've treated the tissue. We've looked at 850 samples from 8 different clinical settings. It absolutely has no bearing on how the tissue was treated beforehand within a certain protocol, how the sample was prepared. The thickness variation might be a little bit within, again, a certain protocol. And what we can show here is the sensitivity and specificity are now approaching better than 99% each. There is nothing in biomedicine that approaches this, right? No immunostaining, no kind of microscopy, nothing whatsoever at this level of accuracy, okay? So now we're reaching a point where we're not just being human competitive, because you can do this much more rapidly than a human can sit down, and even if they were playing video games, right? If you had this on a Wii screen and they were just going bang, 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 right? You can't do it this fast, okay? So speed and accuracy are now trumping human ability, certainly in this field. They're trumping our biomedical abilities even in some way, okay? So I hope I've convinced you that at least in some areas, the chemistry plus imaging is perhaps better at this point than we've ever had it in this case, okay? So let's take a look at exactly how this is done, and then I'll come back to some of these ideas and again show you how this merging of chemistry and spectroscopy perhaps helps out. So just to give you, again, we'll go through a little bit fast on this, and I'll try to refer back to Brad Deutsch's talk the other day and try to take you where things were before. Our system pretty much used to look like this. There are multiple, and perhaps you've taken a tour of my labs. You would have seen some other implementations of this. But classically, the way this work has been done is you take a broadband source. This is just a heated wire, right? It's nothing else. You heat it so it emits light. It comes out. We couple it to a Michelson interferometer. Notice that this is a completely symmetric Michelson interferometer. We don't actually put the sample in the reference arm like in OCT, for example. And you basically encode the spectral information now in time, and you take the light out, put it through your sample, and run it through your detector, okay? So here the encoding from the interferometer is not in depth, it's actually in time. which is a little, it's actually in spectral domain, which is a little different from OCT. Then you can, of course, do a Fourier transform, take a ratio against an appropriate background, and you end up with absorbance, right? Now, the brilliant part of this is Beer's law tells you that the absorbance at any given frequency is directly related to its molecular composition, right? So lo and behold, using light, you've actually detected molecular composition of the tissue. Right? There are two things of interest here. Number one, this is an infrared spectrum, so you see the wave number here. We'll discuss this. Each of these peaks have a very specific meaning, as you saw in the other day in the talk. These sharp lines arise from absorption. This broad sort of sloping background arises from scattering, right? So this is the exact same thing you would see in light microscopy, for example. At the edges of things, it appears dark because light has gone away from there, from those particular pixels, and you can, in your camera, you record those edges as dark. So you see exactly the same effect here, but instead of seeing dark edges, you would see a sloping baseline in the spectrum. So we can extract out all the morphology and all the chemistry together, actually, from this data, and I'll show you how we're doing that. We've done a fair bit of multiplexing in this, and this idea will come up again. But what we get now are imaging data sets that look something like this. So you have X and Y, and in the Z domain, you have spectral data, right? You could have entire spectra that look like this, or you could have something that we coined the term and we developed this first, called discrete frequency infrared imaging, and which just means you collect one frequency at a time, but in a way that you can recapitulate this spectrum in some way. So Professor Mindo, who just walked in the back, he and my group have done some work into which frequencies should you actually collect, right? Because now that becomes a crucial point. Now you have this capability to do this. So now that you can acquire imaging data, what do we do with it? Oh, before I go there, let me just give you one more point of sort of reference. So this is just an interesting side story, but when I began as a grad student, if you wanted to image 1 centimeter by 1 centimeter area with certain signal to noise ratio and certain spectral resolution and so on under certain parameters, it would take you 7 years to image 1 centimeter by 1 centimeter. Okay? Now in my lab, we can do that in about 20 seconds. Okay, so over a sort of almost 15-year, 17-year period, this technology has really dramatically changed. And the only reason why this appears as a straight line or a steady march is because this is actually a log scale. So you have to see how rapidly this thing actually has changed quite well. We sort of stopped counting a couple of years ago because the technology fundamentally changed, and I'll show you what happened there. So it's not the same thing now. We can do different things with the same sources and so on. But really the trick today is how do we turn this data into knowledge, right? That's what we're really after. So for this audience, and I think for your interest, what I'll try and do is explain, first of all, what is the knowledge that we're trying to get? What is the sort of fundamental basis in generating that knowledge? And then we'll talk a little bit about how we turn that using computer programs into something useful. Actually, I misspoke. I'll talk to you about how we do it into something useful first, and then we'll talk about the knowledge generation later, perhaps. The way we do this is we take our data set. So we acquire a lot of data. We have on the side sort of molecular biology experiments, right? So we do some basic cell culture experiments, some basic tissue culture experiments, and then we say that only these features are important, right? So this is a biology-informed sort of feature down select. And we go from this large set to a smaller set. Then we do a statistical pattern recognition approach, where we then do a purely signal processing approach and say, if I were to take one feature, how well would it classify something that I decided was my model, and how well did it compare against the gold standard, right? So we mark off a few pixels every sample, and we say, we predict the whole thing, and we say, how did this compare with this, right? If it compared well, based on the statistics, we say, well, that feature is useful, let's keep it, right? And then we go through and do this one at a time. Then we start combining features in what we call a greedy search algorithm, right? So we take one, we add the second one, did it improve, did it not improve, and so on. So we can, by that sort of process, we can go and figure out what are the real underlying structure in the data that we can use for classification. Okay. At the end of it, then we try to break it. We try to go back. We say, what if we removed this thing, like building a little Jenga piece? We start removing things off and on. And as soon as the structure falls down, then we say, well, this was perhaps crucial for this. And then we go back and repeat this, right, all over again. We repeat it for pairs. We repeat it for things. So there's a fairly involved computational method here. And this is the reason why it takes us to go from acquired data to a full-fledged algorithm. It takes us several months of development, maybe 6 months to 9 months, maybe to a year of development, because we really only produce or only publish or only show you things that we've really validated in this way. If you don't do that, the danger is you have a tremendous amount of data, right? You can spend all day and extract all kinds of information from it, right? And it'll work on one data set. The minute you go to the next data set, it's going to fall apart, okay? So most of the studies in this field actually suffer from this problem. Unless you go through this rigorous process, you really cannot end up with something that actually works all the time. The second thing we actually do, or we take two different approaches, is we validate our results on fairly large data sets. I don't know if you've seen this before, this sort of tissue microarray concept. Back when we started doing this work, I built the first tissue arrays by hand. So we take a needle, put it in a block of tissue, pull it out, and stick it in a paraffin block and then cut those. Now this is done by robotic arrayers, right? So you go in and robots can make 1,000 samples per glass slide or something like that. The first ones we made were 16 on a glass slide. So this technology has also come a long way. And I'll show you towards the end of the talk how we've built systematically heterotypic cell culture models. So we try to build a cell culture model that mimics what's actually happening in the human body to whatever extent we can in the lab, right? And then we use these as inputs again. So both of these things, this is sort of a top-down approach, this is a kind of bottom-up approach, and both these merge to tell us are the data that we're recording, what is the basis, what happens when something changes or not. And you really have to spend a fair bit of time in doing this. But the third thing which you might be interested in is you can go to theory and use that to tell you that if you had a certain structure, what would the data look like, right? And it turns out you need all three actually to really make any sense out of the data and really extract data in some interesting way. If you use any one of them, it's not as powerful, obviously. The second challenge that comes in this field is really trying to figure out which problems you can attack, right? So is this technology amenable to all sorts of things? Could we do pancreatic cancer with the ease that we were able to do prostate cancer, for example? Could we do lung cancer with this technology? And there's one way to do it is, of course, get lots of samples, do this large study spread over 3 or 4 years, try this out, and at the end of the day, it doesn't work, so you go to your thesis defense and say, well, it didn't work, and there could be trouble, right? So there's perhaps a better way to do this, and the way we sort of tackle this problem in our lab is we've built up essentially an ANOVA model that tells us where the variance is, okay? So we can then figure out, is it a problem worth doing? If it is worth doing, then should we spend our time doing it, and under what parameters, what sorts of experimental conditions should we do that? The second thing which you saw a little bit on the first slide is we're sort of coming at this with a completely independent technology, and molecular biology has built up this huge sort of knowledge base. So how do we relate the two, and can it even be related? The third thing, I think, obviously this is from a photonics perspective, this is sort of standard operating procedure. You always try to increase speed and resolution and information content as you have. So there are three sorts of technologies, and I'll show you how they are all sort of related to each other. This is just the ANOVA model description, so if you're interested, we can go over what really happens here. But here's the money slide. It turns out what happens is if you acquire the whole spectrum for something, and once the results were in, we said, yeah, of course, that makes sense, right? It turns out that every frequency has an individual mind of its own, right? So every frequency or every spectral feature is influenced differently by different conditions. So there might be some spectral frequencies that have a wide variation, between person to person, right? But they are, so you can't really use them for disease diagnosis, right? There might be some that have a very low variance between person to person, and those are perhaps useful in figuring out if there's physiologic difference in the tissue or not, right? So again, those things are doable. It turns out, quite surprisingly, we were a little bit surprised that we ended up with about 30 frequencies from this. So okay, so this 2,000 free feature set, from it you can build multiple features and all that. It turns out only 30 were informative, right? And then we went back and looked at the old data, and when we did our prostate algorithms, it turned out we got only 17 that were informative, right? After that, we pretty much found no more benefit to doing those kinds of things. So this number of 15 to 20 features is quite interesting, right? Because it tells you that perhaps there's a fundamental limit why people cannot go beyond a certain point. Now, if you notice, the physiologic difference, though, persists for quite a bit longer. So there should be, in theory, there should be at least 100, 150 features, right, which should give you better information than the biologic variability. And now what you could do with those 150 features is combine them in clever ways to offset biological variability, right? So comparing, let's say even taking pairwise combinations of 30 versus pairwise combinations of 150 just as an exponentially different sort of design space, if you will, right? So what is going on? What is this error that is starting to dominate after a certain point? And it turns out in this particular case at about 20 or so, you start getting this residual in the ANOVA model, right? This is the unexplained error that we found in the ANOVA model that doesn't allow us to go beyond about 30 features in the infrared data. Right? So this was extremely surprising because we had accounted for every single, we were very proud of ourselves. This was the best study design we'd ever done, accounted for patient to patient, clinic to clinic, everything that we could possibly think of. And yet there was this residual that didn't make sense. So we did this study in 2006 or so. This curve, this graph was obtained in 2006, and it eventually got published in 2012, right, 6 years later. And what happened in the meantime, we realized what was going on, and we figured out why we were getting this residual and how to get rid of it in that sense. So to understand this residual, understand how we're doing spectroscopy, right? And this is really some very, very trivial stuff. Again, once somebody explains this to you, it makes perfect sense. It didn't until that point. So the way we're doing it is we take the intensity recorded by the detector without a sample first. So this is the electric field squared transmission of the instrument. It's basically mapping the source as transmitted by the instrument onto the detector, right? Intensity-wise. Great. Now, in this, we put a sample in the path. And the only thing Beer's law tells you is that the sample absorbs light, which is an exponential decay in intensity, which is right here, this term, great, based on the path length of the sample, and this should be it, right? But there's also this transmission function that we have put in because of putting the sample in, which is not chemical, right? It's just perhaps scattering or whatever. And then when we calculate absorbance, it turns out this is Beer's law, just this first term, right? So this is 150 years old, well established, confirmed by everybody, everyone agrees with it. But what we have also is this very small term. And what textbooks don't tell you is that hopefully everyone says this is small, just sort of get rid of it. The transmission functions are equal, so you'll get 1 and this will go down to 0 and so on. But it turns out when you do microscopy, this term is not really small because you have scattering, you have diffraction, interference, you have all kinds of effects. So what we have done is really modeled the microscope from, I don't know if did Scott Carney talk about this at all a little bit in his talk? Okay. Otherwise I can go over it again. What we have done now with Scott is modeled from source to detector the entire image formation process in a microscope using a couple of ways. And I can show you both those ways. So I'll go over this, I think, fairly quickly. The only interesting point, which again, stop me if you've heard this before, but the only interesting point is what we are interested in is the absorbance here, right? We're interested in these sharp lines. It's a symmetrical wave sort of form around this band. And it's the imaginary part of the refractive index. That's what the absorbance depends on. Right? Unfortunately, it turns out that the imaginary and the real part of the refractive index are related. And in the mid-infrared, you get this sort of anomalous dispersion idea where you have very rapid, almost violent changes in the real part of the refractive index when your absorbance set as the maximum. Right? So precisely at the point where your signal-to-noise ratio is the highest, you also have the highest confounding effect. Right? Light is absorbed based on this, but it is transmitted based on this. Right? So light might have been absorbed correctly, but then because of scattering or because of mismatches in the real part of the refractive index or all the other factors that go along with it, it actually went somewhere else. Right? So you didn't actually see light from where it was supposed to arise from, and that's really where the transmission function of the instrument changes when you put the sample in. Okay? And that can become quite large. It can actually become larger than the absorbance that you want to record, and that's the problem in this field. Okay? So I think did Scott cover this properly? Anyway, let me bring you to the results, right? So what we've been able to do is take, yeah.
So then take the transmission function of the, other than the optical transfer function of the system?
Yeah, let me clarify this point, right? So when it's not the physics-related optical transmission function. Right? It's not the physics function. When we say that what we're doing essentially in spectroscopy is measuring transmitted light, right? We're not measuring the light that's absorbed by the sample. We're measuring what's left, right? So that's the transmission of the instrument in that sense, right? So it's not the optical transmission function. It's the transmittance of the instrument, to be more precise. So what we've been able to do now lately, this is the new thing, is if you have, you can take the model and you can invert it back. If you have some knowledge of the spatial morphology of the sample, right? So in this particular case, this is a sphere which is very, very complicating because your sphere is of the size, is of a diameter that's exactly in the mid-band of your spectrum. So now you're getting scattering all over the place, and you get data that doesn't look like what it should look like. But again, long story short, if you were to take, this is PMMA, and if you were to take PMMA spheres of different sizes, essentially you get spectra that look very different. And again, this is valid in infrared or in Raman or in whatever technique you want to use. But what we can do now is with the appropriate methods, we can recover back not only the morphology of the sample, but also the refractive indices at every point in that case. For simple structures, this is possible right now. For more complicated structures, one has to think of other ways to do it, which is still undergoing some ideas in the lab. The way we've tackled this, again, just to give you a brief idea, is now to give you an idea of the complexity of the problem, is the way you make your focus in the microscope is you take a plane wave and you just add it up in the angular frequency, right? So that's one frequency. But the wave number frequencies or the spectral frequencies are independent of each other, so you consider one frequency at a time. So that's your second frequency, right? Now if you have a 2D sample, so these are thin samples, remember thin compared to the wavelength in general. So if these are thin samples, then you have two spatial frequencies. Of course, you could have a third spatial frequency if it were a thicker sample, but you have two spatial frequencies now. So there's 4 frequencies that you have to be aware of, and at least 3 of them have to interact. You can take the wave number to be a separate frequency because wave number you can just loop over. But the angular frequency has to interact with the 2 frequencies, right? So it's a coupled system of 3 frequencies and a loop of a 4th, right? So you have 4 loops with 3 of them coupled. You can imagine a single frequency takes a day to run a proper simulation in this business on a desktop PC, right? So if that's the forward model, trying to invert it in this way in spectroscopy is just not going to happen. So we've taken some approximations. I don't want to quite get into those details, but we've taken 2 approaches. One is we have the largest, or at some point it was the fastest supercomputer in the world right here on campus. I guess it's not anymore. So we've tried to work on that system. Of course, it's not a very practical idea. It's just to solve equations. The second is we've taken some clever sort of mathematical shortcuts, and I think I'll skip that today because we don't quite have the results worked out yet. But if you come back next year for the summer school, I think we'll have it ready, especially those of you who fail and come back. But I'll show you some insights that have still come about, right? So we can take a few frequencies, so this is about a 20-day simulation that my student actually did. And what we were doing here was trying to answer this question that, okay, you've got a sample, you've got all these frequencies, they all interact, you get some image that comes out from here. That's great. But what we didn't sort of understand, again, for about 15 years in this field, was when this signal comes out, right? Think of it this way. Let me explain it in the most sort of intuitive way. So think of an analog signal that has a certain frequency that's coming in, right, from the condenser or this thing downstairs. Then it comes through your sample. It picks up a certain set of Fourier frequencies, which is the scattering frequencies from the sample, right? So it's related to the frequencies that are present in the sample structure. Now the top lens acts as a bandpass. So it takes this whole frequency domain, cuts it off at a certain point, transmits it over to the detector, right? Now if you had a photographic film with infinitely fine detectors and you put it there, you would see all of them, right? But our detector is actually discrete. It's a CCD type detector, right? It's a bin detector in that sense. So you're taking a certain spatial region and you're binning it down, right? So this is a very trivial question. What should be the spacing of the detectors, the center-to-center spacing of the detector? It should be twice the highest spatial frequency you get, right? This is absolutely trivial, first principles knowledge. Right? It turns out that in the infrared domain, that frequency turns out to be 1 micron. Okay? If you think about it, the central wavelength in the bandwidth of our measurement is about 5 microns, right? So if you look at Rayleigh's criterion, it says, well, you've got 5 microns, you've got a 0.5 NA lens, you're probably at a 10 micron resolution. So all the instrument companies said, okay, I'll make my pixels 5 microns. I'll give you a little extra over there, and you've got that. But it turns out if you do this analysis, you're at 1 micron is the answer you get. So what's right? Is it 1 or is it 5 or 10 maybe? Is there more information beyond 10? Is there more information beyond 5? Is there more information beyond 1? So of course, the easy thing to do in this particular case is just use a higher NA lens and compared to a lower NA lens, which is done in that case. So here's the result, and I think, again, this should prove it very clearly. Here's the simulation under different conditions, and this is the commercial system that was there when we started doing this, right? So this is a 0.45 NA lens, 15x magnification. It gives you about 5 micron per pixel at the sample plane, and the images look like this, right? This is a slightly higher NA lens, so we're cheating a little bit here in this comparison. Slightly higher NA lens, but it's got the right magnification to give you a micron pixel, right? So this is our best, this is the best commercial, right? This is, you can see that right now. Now, if we did it right, if the commercial system was designed with this criterion and not with Rayleigh criterion, for example, you would get an image that looks like this, okay? So it's not obviously as good as with the higher NA lens, but if you compare the commercial with what we are proposing, it's still much better, right? So there is life beyond Rayleigh in this sense in chemical imaging. And the reason why there is life is because if you think about Rayleigh's criterion, again, we can have a 1-hour lecture on this. Rayleigh's criterion says the minimum distance between 2 point sources and so on, blah, blah. But what it doesn't say is that the 2 point sources have to be spectrally identical, right? If they are not spectrally identical, you can actually locate the centroids of those 2 point sources to infinite precision if there is no spectral overlap, right? If there is spectral overlap, then there's a very complicated relationship between resolution, localization, signal to noise, and the amount of overlap, right? This idea of separating overlapped signals in spectrum is actually quite similar to the idea of separating overlapped signals in time, right? So if you've heard of Palm Storm, the sort of super resolution fluorescence microscopy systems, what they do is they excite fluorophores, but only one is activated in a focal volume at one time, right? In the next shot, another one is activated in the next shot. So if you had things that were spectrally distinct, you could do one-shot super resolution, okay? So what you're seeing here is midway between hardware advances and the software advances needed to do super resolved spectroscopic imaging. So that's where we're headed now. The hardware is implemented. This is now commercially available from at least Agilent, and I think PerkinElmer will introduce it later this year in this case. But now you can do what is called high definition IR imaging now, just for the heck of it. It's really higher NA and better pixel spacing. That's really what it is in that sense. So the way this has really changed the field in the sense is this. So this is commercial, best state of the art. This is what we get. This was the first study we did. This is actually on a synchrotron because now you have a much smaller pixel, so you need higher light throughput. But we've done some things in the lab, and this is stuff we can do in the lab now. So here's colon tissue. That's the best commercial system available. This is what we get routinely in the lab now, right? So things are looking much better. You can actually start to see subcellular features. Here's the nucleus. Here's cytoplasm. It's a single cell, and you can see things much, much better. So it turns out in chemical imaging, or in infrared microscopy, the wavelength's longer, but the quality of data, the 1 micron pixel, is precisely the sort of right pixel size you need in pathology. You need about 300 nanometer to a micron resolution. That's very sufficient for most things. It's 100x. When people say 100x, that's what they really mean, that kind of resolution. So it turns out we can actually get that information now using wavelengths that are actually much longer. This is very counterintuitive in some ways. The way we've done this, and this is, we worked with a company in San Diego for about 4 or 5 years. We got the first quantum cascade lasers from them, and these are narrow bandwidth, single frequency lasers now that we can use that are very, very high intensity, that are 1,000 times brighter than the heated wire source, okay? So we can use these in intelligent ways, and there's some issues that come along with using lasers, but we're trying to resolve those. So this is absolutely the next frontier. So watch out for this paper. There will be lots more coming in this space. No one's published anything with a quantum cascade laser for about 1.5 years now, but I suspect by the end of this year there'll probably be 100 papers on this topic. There are at least 2 commercial manufacturers now who are making this sort of instrument, so this should be much more viable. So I think I've got maybe 10 minutes or so, and I'll touch on 2 other issues. One of them is this sort of side idea, and this is just relating back to your lab. We have a whole molecular biology effort in the lab in which we try to figure out what molecular biology of heterotypic interactions in cancer, which means dissimilar cell types talking to each other in cancer means, and how we can analyze that. And the whole idea behind all of this is really what you have to think about when you analyze tissues or look at cancer progression in particular, is that there's a chemical change, of course, but there's also space and time, right? These are two other variables in this. And there's also hierarchical order in organisms. So something happens locally, right, at the gene level, then it transfers hierarchy to a protein level, which then transfers hierarchy to the cellular level and so on. But even on the gene, something happens at different points in the genome, at different times on the same points in the genome. Different kinds of chemistries might happen on the same type. It could get inhibited, it could get activated, whatever, right? So if you think about the whole information information content, ideally what you would like to do is have in all space and time, all chemistry at all places, at all orders, right? Of course, it's not possible. No technology will give you all the chemistry that's there in this place. So what you're left with is asking the question that how many hierarchies do we need? How much chemistry do we need? How much space-time should we include into this model, right? So we have now an approach that we call systems pathology in our lab, and we're just about ready to send the first paper out in this case. We've been working on it for about 6 years now. This is called, in this particular case, what we're trying to do is trying to get information from different places, couple them in such a way that you get a better diagnosis than any single place alone. Okay, so that's the idea here. And I'll just sort of skip over. We talked about this. There's a sort of bottom-up approach and a top-down approach, and that's what we're doing in this particular case. So the way we do this is, let me quickly explain this, is we started many years ago doing cell culture of single cells laid down on a layer of, on a line of fibronectin, right? Then we went to 2D cultures. We started culturing skin in our lab many years ago, but this is commercially available, so we just wanted to meet the skin model people. What commercially they don't do is put melanoma in here, so we would stick melanoma in, watch it grow over time and so on. Then we slowly built up capability over the last few years, and now we routinely do these kind of things in the lab that we call spheroidal co-culture models. There are two challenges in this. One is how to maintain the spheroid, how to maintain a mass of cells with the right sort of shell morphology in it. The second is how to introduce culture conditions that you can put other kinds of cells in it, and it still sort of looks physiological. Again, it's a cell culture, so it's not very true, but it recapitulates some of the interesting things. What you perhaps saw downstairs in the lab was the 3D printer. So the eventual goal is that we'll not just make this, but we'll actually print a whole sort of microstructure of a piece of tissue for you, right? So you would take cells from a patient who's diagnosed with cancer, take them to your lab, take those cells, culture them, propagate them a little bit, put them into the printer and print out different tumors for that person and see which drug would work, for example, on that person or not, right? So we think we can do this in reasonable time. This is just recapitulating what we've done with the 3D printer, but essentially the way we're doing this is you look at this, and this is really the key behind this. We can use our imaging techniques to try and figure out some of the rules of organization in the breast, for example, put them into a computer program, and we can generate a whole human breast at 10 micron resolution fairly rapidly now in a day or so. In this case, it takes 5 terabytes of storage, but that's a different issue. So we can generate this. We can generate an artificial thing, go in and simulate this. We can say, what would this particular breast that we've generated look like under CT or under ultrasound or under what have you, those kind of things. Stick a needle in. Again, this is all computational. Stick a needle in, pull out a piece of tissue, and then go and print it, because that tissue forms the sort of map for our printer to move. And then we can go and print that. In this particular case, this is just a toy sort of example. What we're showing here is this bar is 1 millimeter. So what we're showing here is that we can actually print trees that go down from millimeter-sized trunks and to this point, I don't know if you can see it very fine here, but to about 50 micron features quite easily. 50 micron means we've got about 15 micron of control on how we go about doing this. If you look at commercial 3D printers, they have about 1 millimeter of control or so on. The next thing, which is quite interesting, if you spoke to the student who was downstairs, is it takes us exactly as much time to print this with a 50 micron feature size that it takes to print this on a commercial printer at 1 millimeter size. Okay, so we're very, very fast downstairs, and we can do this quite rapidly. We can do other things, but I just want to point out what we've done here. We can print trees, so I can make you 24 identical looking sort of breast scaffolds. We can put them in, grow breast cancer cells on these, and you'll have identical 24 breast cancer samples to do drug discovery or what have you on these, or imaging if you're interested. This is now online, so I won't explain this. But what we've been able to do, I'll just explain this curve. What we've been able to do is figure out the heterotypic interaction between breast cancer cells that are just starting to proliferate and fibroblasts with a set of 46 proteins. Now it turns out when you take those 46 proteins and find their corresponding genes and you do the comparison against public databases that are published, it turns out we beat PAM50, this is PAM50 results, which is currently the clinical test that one would use in breast cancer, right? So we can better separate out those who have dangerous breast cancer from those who don't compared to PAM50 as of right now. The interesting part is that this is based on the gene signature of the primary tumor. Our signature is based on the interaction of epithelial cells with fibroblasts in this particular case and with another cell that we're putting in now. So in this particular case, what you might be able to do a few years down the road, what we think we might be able to do, is put these 40 or 50 proteins on a chip. You could take a drop of blood from somebody's body and put it into that chip, and you would get a readout of these 46 proteins or not. So instead of the person going in for mammogram every year where they try to figure out whether there's a lump and what that means and all this business, you just do this blood test once in a while, right? You do this blood test, your annual checkup. It doesn't tell you if there's a cancer there or not. It tells you if your cancer is there and it's growing. Okay, that's the trick here. So this is the way, and with this kind of approaches, we can really go back and take. This is a very, very long, long development process where it may never even pan out. I'll give you, that. But if you come back to the summer school in about 20 years, I hope this will, we'll have results from this. But again, this idea of going from cell culture to analysis to actually finding something, it's multiple steps of failure in the middle, but this path will now go down, and this is established now. So where does spectroscopy or photonics actually come in all of this business, right? You're wondering now. So the trick in all of this, the reason why this worked in our lab was what we did was we put the epithelial cells and fibroblasts next to each other, and we watched it without dyes, without labels, but using spectroscopy, right? So we didn't know what was changing, but we knew nothing's changing, nothing's changing, nothing's changing. Now something's changed, right? And then things keep changing, whatever. So we focused down on that time window where just there was this massive change in the middle, and that's where we obtained the time to analyze it. And that's the signature that we've actually obtained. So if you were doing this without using infrared spectroscopy in your lab, if you were just a molecular biology lab, you would miss that very small window. You would perhaps be over here where lots of changes have now happened, or you might be over here where you say, well, there's no influence, right? So there's a very narrow window that you have to get this right in, and that's really the trick why this works. We still have to prove it, but I think why this works, okay? So that's the reason perhaps why people missed it before. This is a little bit, I think I'm going to run out of time in another minute, so I'll point you to two other things in the lab. The first is, again, as I mentioned, we are very interested in trying to get more and more sensitive sorts of information because we would like to detect these proteins, these 46 proteins in the blood at some point, right? So we have an intrinsic motivation to do that. And I think Brian Cunningham spoke about this as well. How do you make what he calls photonic crystals and put things on top of those? We have a somewhat more modest name. We call the surface enhanced Raman spectroscopy substrates. But essentially it's the same idea, is how do you couple light into a localized spot where you place your antibody and then the protein comes and binds to that antibody. The way we've done this is, of course, built a surface, but then we've also built the underlying infrastructure to perhaps have a standing wave in some case, so you interact multiple times, to illuminate from the bottom and the top. In some case, you have interference, 2 photons, so you can localize things at a certain point. And again, this is an ongoing story. I don't think anyone's got a good solution yet. On the other side, we've also made probes. So we can take a nanoparticle, go stick it to the protein. So you've got a one-shot label, right? So you have an antibody to the nanoparticle. This goes in. You don't have to stain it again. There's no secondary antibody. You're not restricted to, say, goat and rabbit and so on for one thing. You just use it once, and this works all the time. This is, again, much easier said than done. So we've spent a number of years as well doing this. But again, these are directions that you need to pursue. And again, the track we've taken in this is that we hide all our sins of the nanoparticle in the way we do spectroscopy. Okay? So since we know how to do spectroscopy intelligently, we can then design a nanoparticle that perhaps is not as good, but you can make use of it anyway in some clever ways. Okay. So again, if you're interested, the references are there. The last thing I think I want to touch upon is this. I've got maybe 10 seconds more, Steve. Is that okay? The last thing is this idea of systems pathology, right? So the question that we talked about, how much information is enough? What should we look at? What should we not look at? And so on. How far away from the tumor should we be looking at? And things. So again, this is there's actually an update to this. This is Bob Weinberg's paper in 2011 in Cell. When I made this in 2006, his paper was not out, but in 2011 he has a similar figure out. I don't claim he stole it from me, but obviously this idea has been around for 100-some years, right? So the problem is, everyone says the microenvironment is important, but how are we going to measure it, right? So you can take immunostains, which we just discussed about 40 minutes ago, and we said they weren't very reliable, right? So you could take immunostains and you could stain the thing, but now you're going to stain it 5 times, 6 times, 10 times in the same tissue, and each one of them is only reliable 80% of the time, right? So 0.8 raised to power 10, quickly somebody do that, it's a very small number, right? So getting quantitative information from staining repeatedly to analyze the microenvironment is not a good idea. You just cannot do it, right? So you can multiplex quantum dots, you can do all that sorts of stuff, but it's not really, there's no real technology to look at the entire microenvironment on its own, right? So if you remember the second or third slide I showed you is that we can find cells, quite precisely. We can find cells better with more accuracy than anything out there. And we can multiplex it all at the same time, right? So if let's say our specificity and sensitivity is 99%, right? It is 99% for all the cells together. It's not 99 to the power 10, or it's not 0.99 to the power 10, which would again be a small number. But it's 99% throughout. There is nothing like that that allows you to do microenvironmental analysis. So our technology, we can instantly measure all the cell types in the microenvironment, right? Great. Now the tough part comes in. Can we actually measure the transformations in those cells, right? So finding where they are is not, we can do that, fine, great. But are they changed? Are they changed in response to the tumor, right? Which is even bigger question. So one can do that, and I'll cut again this long story short. What we've been able to do is take a set of patients and show that this information that we add in is independent of the current prediction methods. It exceeds them. We can address the midpoint of prostate cancers. We can address whether cancer will come back or not. I think this is all interesting. But more interesting is, where's this information coming from? And we can go back to the microenvironment of the tumor and figure out where this information is coming from quite precisely. We can then make better predictions based on what this actually tells us. We can perhaps go in and do genome sequencing here and say what really is changing and so on. So this is still an ongoing story, but at this point, what we've been able to do is bring up a risk score, right? So if you're high in there, so again, remember, these are the 70% patients right in the middle where we can't do a good job. So this is the clinical gold standard 1, and 2, this comes from the East Coast, this is the West Coast, so it's like RAP, it's competing schools of thought, right? But neither one of them give you a very good, prediction, right? What we can do is not only give you a linearly changing prediction, but also a 20-fold compared to 1. So if you're in this group, go in for surgery really quickly. If you're in this group, definitely, wait at least for some time. It's not sure that you don't have a lethal prostate cancer here, but you can afford to wait for a year or 2 and see how it goes, right? So I think we're clinically actionable as of right now. With that, I think I'll just stop here and just this sort of recapitulates things that I talked about. And if there are any questions, I'll be happy to take them. Thanks. Yeah.
Is it possible to use just algorithm to reduce the dimension and get the same?
Yeah. So this is so yeah, you're absolutely right. And in 2007 or so, the standard in the field was that people would just use PCA, right? Would just do principal components analysis, transform the whole thing, figure things out. And when we wrote our paper in 2007 saying that's idiotic, the reviewers were not happy at all. I mean, they just beat down on that thing and said it's not possible that you're taking information that is not contained in the PCA. And the argument was, when you do a brute force thing like that, you don't know which information you're including, right? Is it the scattering induced information? Is it the chemical information? Is it that? And that's the problem. So if you knew what your input was, if you knew what your variability was, then you could assign the variability into the appropriate bin, like you would do in a PCA, for example. But I personally don't think just statistical processing is enough.
The other thing is resolution, because you can go beyond the diffraction limit.
Ah.
Using the spectral features.
Yeah.
Using the 2 pixels, typical features. Then won't it give the disparity of resolution across the tissue?
That's brilliant. So so what she's saying is that there are a few things, right? So one is if you have a domain, the resolution within that domain versus how sharply you can delineate the boundary will be different, right? That's a good point. The second thing is if you use different frequencies, right? So if you use 10 micron versus 2 micron wavelengths, you'd get different results, obviously, right? Because so the first part, if it's within a domain, if it's chemically identical within a domain, you really don't want to separate that domain. So you don't care that within the domain. You only really care about the edges, right? How sharp you can make the edges in this case. The second question is really a limitation, and you have to design your optical system to go to whatever side you want. So we stick to mid-band. You could go to the short band side. You could go to sort of the long wave side and design your instrument. When you do, but that's just the hardware, the data you get back. You can do sort of deconvolution approaches. I just call them deconvolution in some way. You can do physics-directed or just regular deconvolution. It turns out that you're going to turn everything into a smooth surface if you do that anyway. And so, the value of that is, again, questionable at that point. But just purely, do you get more information by changing hardware? The question is, till 1 micron you do, and then you don't beyond a micron. So you can't get 100 nanometer resolution just by hardware. Even if you made the pixel size 100 nanometer, you're not going to get anything.
Some of the actual 3D printed models, right? Do you incorporate some of the mechanical properties?
So not yet, Steve. The trick is how to control collagen cross-linking, right, in 3D. And the best methods we've found are people say, oh, mix it quickly at 37 degrees and put it in the fridge at 4 degrees and keep it there for 27 minutes and then bring it out and blow on it and then put it back for 2 more. I mean, it's just a recipe. So collagen is so variable that I just don't think you can do it with collagen. With synthetic polymers, I think you can, and there's well-known ways to do that.
Any other questions? Okay, very good.
Okay, thanks.